Okay. Hey, welcome back to the Classics in Immunology Journal Club. And this week we are in 1991. And before we get started, I just want to remind you to like the video on the YouTube site and click on the bell to get notified for more, subscribe to the site, and check out my social media things. Scroll down on the YouTube site and you'll find my webpage. And then on the webpage, click on the writing tab and you'll find all the classic papers there. So if you want to check the veracity of my presentation, you can read for yourself exactly what's going on. So today, we're doing a paper authored by Hubert, Schorle, Thomas Holtschke, Thomas Hiinig, Anneliese, Schimpl, and Ivan Horak, all from Würzburg, Germany, University. And the title of this paper is The Development and Function of T Cells in Mice Rendered IL-2 Deficient by Gene Targeting. Now, the background in this paper, is that there were just almost immediately, two previous reports of IL-2 deficiency or lack thereof in humans, one from 1989 and another one from 1990. And so this is 1991 where they did this. This was the first cytokine knockout in a mouse where they knocked out the gene. And that had only been really reported in 1989. And so these people were right on top of the latest technology of the day. And the human IL-2 deficiencies, the first from 1989, had severe combined immunodeficiency, meaning they had deficient T cells as well as B cells. And the patient was an infant, essentially, and was treated with bone marrow transplants ×2, both of which failed. And then they treated the patient with recombinant IL-2, and that was a success. The second report came out in the New England Journal in 1990, and that poor patient also underwent 2 bone marrow transplants, but died of GVH disease after the second one. In both of these instances, it was not a shocker because everybody expected the patients to be immunodeficient because IL-2 had been touted by myself and others as the be-all and end-all of immunology, and that you really needed IL-2 to cause the clonal expansion of antigen-selected cells a la Burnett. And if that didn't occur, then you would not have immunologic memory. And we talked about that last week. And you would not be able to ward off infectious diseases. And so you would ultimately succumb to severe infectious diseases. Now, Ivan Horak is the senior author on this paper, and he was a pathologist, and he was anxious to do this gene targeting approach to knock out a gene, and he wanted to knock out an important immunological gene. So he went to his colleagues at University of Würzburg, Anneliese, Schimpl, and Thomas Hiinig, and asked them, so which gene do you think I should knock out? And they both chimed in that they should knock out the interleukin-2 gene. And so that's how it got to be the first lymphokine, cytokine, interleukin gene that was knocked out. And this paper turns out was a game changer. And so we'll see what the data look like when we get into this thing. In the introduction of this paper, they go through the importance of interleukin-2 and the immune response. And they said that, all of the things that have been done so far with IL-2 had been done mostly in vitro, and they wanted to have a system where they could study what would happen if you didn't have any interleukin-2 in vivo, and in this case, a mouse, because that's where the technology led one in this instance. And so, in the materials and methods that I'm not going to go into the intricacies of gene targeting, but what they did was they inserted a neomycin resistance gene into the third exon of IL-2. There's 4 exons. It's a small protein, 15,000 Daltons, and so from a genetic standpoint, a molecular genetic standpoint, it wasn't a huge problem. I mean, there wasn't a huge gene that they were trying to knock out. And so that's the background. And so we need to go into the results now and the screen sharing. And this is figure 2 from this paper that was published in August of 1991. You know, if you read this figure caption carefully, you can't figure out which is which, but these are normal mice in the upper cells from a normal mice in the upper panels and from the knockout on the lower panels. And they simply wanted to know whether or not the CD4 and CD8 ratio in the thymus and in the lymph node were abnormal or whether they were normal. And then they wanted to look at the ratio of the cells in the thymus and the lymph node that had the T cell receptor or against the relative cell number. And if you look at these panels, they're basically telling us that there was no real difference. And the thing about the data, and so that if you look at CD4 versus CD8 in the thymus, most of them are so-called double positive. In the normal and in the knockout, single positives were identical in these instances. And I should say that that all of the data in this paper that came out in Nature were from one pregnant mouse that had a litter of 7 homozygous IL-2 knockout mice. And they waited until the mice, so the mice were born and they didn't die in utero, and this is a good thing. At least they had something to study. And they let them grow up to be 4 weeks old, which is just about, they at 5 weeks, they undergo puberty. And so they're just about adult stature at that stage. And you can see that the percentages of TCR positive thymus, both thymus as well as lymph node, are about the same. And so that was the first figure. And then in table 1, which is up here, this, tables are very difficult. You have to study them. But let me see if I can lead you through this without getting you totally confused. These are the genotypes of the mice. That was a homozygous normal mouse, heterozygous, plus minus, and then 3 knockout mice. They cultured these cells and in vitro, and what is depicted here is the thymidine incorporation over here, and then the IL-2 bioassay from this long-term CTLL lines that we created, which was the standard IL-2 bioassay. And just to get you oriented here are the culture conditions, the medium on the one hand and con A. And I think this was like 48 hours or so. And so what they cultured the cells in medium alone and also added IL-2. This is thymus. Here's spleen, and here's lymph node. And here's con A as a T cell stimulant. So you can see that there was a marked increase in even in the, just this is the normal situation. And the thymus, add IL-2 and even it gets better. And then in the spleen, without any IL-2, there's the thymidine incorporation is markedly increased when you add con A and add IL-2 and it even gets better. And here in the lymph node, it's a similar story. And in the CTLL assay, just taking supernatants from these conditions, there was clearly easily quantifiable IL-2 produced. Now let's just go all the way down here to the knockout. And you can see here, this is medium in the first knockout, very low in the thymus without IL-2, add con A, and there was an increase. That was a shocker. That was a surprise. And then this is adding IL-2. There was an increase without any con A, but in con A it markedly went up. Spleen, same story. And so the shocker here is that these cells could proliferate in the absence of any IL-2. Because up until this point in time, if you recall, IL-2, his original name, which I'd always preferred, was T cell growth factor. Over here, you see that the cells that were knocked out, their gene was knocked out, which incidentally, I forgot to tell you, had no detectable messenger RNA for IL-2 or protein. And here you can see that there's no protein in this instance. And so that's the first thing, is that it was striking that they could proliferate at all, because we had thought, as well as others, that this was the only T cell growth factor. And the other thing they wanted to measure in figure 3 is whether or not these knocked out T cells from IL-2 deficient mice could function as helper cells for the B cells to make immunoglobulin. And so in this experiment, they've measured the number of plaque-forming cells per culture activated by anti-mu in the absence in the top panel and panel A, and in the presence of added IL-2 to the cultures. So when you activate the B cells of the anti-mu and use T cells from a normal mouse, you get 900 plaque-forming cells per culture. Here, the heterozygote, it was just the same, but in the knockout, the mice didn't make any IgM. So that was quite striking. And then if you took it from, if you added IL-2 to these cultures in the IL-2 knockout, it replaced that ability. Now they can make plaque-forming cells and make IgM antibody. And in data not shown, but put into the text, they took serum from these mice at the age of 4 weeks, and they measured the concentration of IgM, IgG, and IgG1, and also IgG2A and 2B in the serum of these mice. And they found, actually, even though they couldn't produce IgM in vitro, they were making it in vivo, and the concentration of IgM in the sera were comparable in both the knockout as well as the normal mouse. And then the surprising thing was that IgG1 from the knockout, about 2,000 micrograms per ml of IgG1 versus only about 50 in the normal mouse. So they had increased levels of IgG1. They also had increased levels of IgG2A and 2B. And so that was also the other shocker. There was something strange about these mice, that they weren't making IgM in vitro, but in vivo they were making it, and then they also were making huge amounts of IgG. So that was really a strange kind of thing. And so in the discussion of this paper, they only made a couple of points, and I can go back to stop the screen share at this stage. In the discussion, they said one of the very most important things that they yet needed to do, they basically took the first litter from one mouse that was the knockout to do the experiments that are placed in this paper. That's pretty good. You get a Nature paper out of only one mouse. What can I say? They were hoping to be able to test the response of IL-2 knockout mouse to infection. And that was one of the next things that they went ahead and did. And they were hoping that these IL-2 knockout mice could provide a model system in the mouse that you might be able to treat IL-2 deficient mice by injecting IL-2, and this could be a good thing for man and God and everybody else. So that's it. That's the paper. August 1991. This paper subsequently, for the next 2 decades, from 91 to the aughts and into the teens, this paper it set off a seismic shift in the IL-2 world and in the worlds of immunology for 2 basic reasons that I've already mentioned. One was the results suggested that there was something else that could be a T cell growth factor besides IL-2. It was, relatively speaking, on the minor level as compared to IL-2. So the IL-2 still seemed to be the principal T cell growth factor. But it also allowed the T cell, the people that were interested only in the T cell receptor as being the all of the end all of immunology. Once they found the T cell receptor, they were happy. They figured that they were going to get the Nobel Prize, and this was the end of immunology as we knew it. Well, they never accepted the fact that maybe these interleukins might be very important in the regulation of an immune response. And so they wanted to be able to say that, well, the T cell receptor set the thing in motion, activated the T cells, and then interleukin-2 and other T cell growth factors just amplified what had already been started by the T cell receptor. But as time went on, that clearly could not be the case, because as people got into the signal transduction biochemistry after the T cell receptor in the first few seconds to minutes to hours and so forth, it was clear that the signaling pathways activated by the T cell receptor were different than those activated by the IL-2 receptor, number one. And number two, the signal transduction pathways and the transcription factors that were activated by the T cell receptor did not cause G1 progression, cell cycle progression, DNA replication, and mitosis. So lymphokines, T cell growth factors, were still primarily important in this whole business, but that took a decade or more, and some people still don't accept the fact that the lymphokines are the primary, or are the mediators of T cell clonal expansion, which of course is the basis for cellular memory, immunologic memory after an initial infection or a vaccination. So it's the raison d'etre behind vaccination as well. The finding that there was defects or dysregulation in the antibody response out of the B cells was unexpected. The fact that they could not make IgM in vitro, but they seemed to make it in vivo, and the fact that they made huge quantities. I mean, this was really a lot of IgGs being made into the serum of these mice. So that really was the tip-off that there was something funny going on here. And that's why I say that this paper created a seismic shift in the IL-2 world and in the immunologic world. And I couldn't figure it out at the time. I was just in the process of moving from Dartmouth to New York, but I got on the next plane. As soon as I read this paper, I decided I had to go meet Ivan Horak and find out what was going on over there in Würzburg, Germany. I knew Thomas Hiinig and Anneliese Schimpl. I'd met them at meetings and talked to them and so forth. And I didn't know them well, but I, they were acquaintances. So I got on the next plane to Germany and I visited Ivan and Thomas and Anneliese. I gave a seminar and we talked things over about, so Ivan Horak was interested because he was a pathologist. He wanted to know what I thought about their results. And I didn't have a clue. I had no idea what was going on in these mice. But you know what? 10 years previously, 1981, when we were working on the intricacies of defining the interleukin-2 receptor. And you remember the story that once we had all the data in from our binding assay and so forth, it looked as though, to me, that this was a real bona fide hormone receptor with all the classic criteria of what the endocrinologists had already established for hormone receptors. And those were that the binding was saturable, meaning that there were only a finite number of receptors on the cell surface, that the binding was of high affinity, meaning that you only need a very low concentration of the ligand of the hormone in order to stimulate a biological response. The binding data and the biological response data were coincident, and that was good from the standpoint of the physiology of the whole thing. And so I ran up to Alan Monk's lab. He was the endocrinologist who was helping us become endocrinologists as well as immunologists. And I said, well, Alan, this is a real bona fide hormone, right? And receptor. And he said to me, he thought for a while, he was very, steady as you go, low key kind of a guy. And he said, well, not really. Because in order to call it a hormone, it's got to be internally regulated. There has to be negative feedback controls over the whole system. Because otherwise, once you set it in motion, we added IL-2, activated the system, it would just continue on ad infinitum without any more things. So there's got to be something turning it off. That was key. And as time went on, and what I did learn from Ivan Horak when I went over there and visited him in Würzburg was that, they had done these first experiments that they reported in their Nature paper on 4-week-old mice. Wow. And it was only the first litter that they had. So as they got more mice and let the mice grow up longer, so that they looked, ostensibly they were fine for 4 weeks. But then in the second month of life, they started to become ill and failure to thrive. They stopped gaining weight. They stopped moving around their cages. They just sort of sat there in the cage. Their fur became very ruffled, and they were clearly sick animals. And they had early data then that these mice were developing autoimmune problems. And there are two manifestations that they had developed. One was colitis. They were having severe inflammation in their colons, which essentially would kill the mice. They would live to be about 9 or 10 weeks old, but then they would succumb to colitis. And the other thing that they got was autoimmune hemolytic anemia. They started making antibodies to their red blood cells, and they became severely anemic. And that was the other thing that killed them. I mean, they just destroyed all of their red blood cells. So that was sort of incompatible with life. At this stage, I don't want to go into all of the things that happened subsequently after this paper, but I would like to refer you back to my book, Molecular Immunity. If you look in this book, the next 20 years are essentially focused on in this book in a chapter that is called Regulatory T Cells, Chapter 8. This is Chapter 8 from my book, Regulatory T Cells. And I review all of the findings that had happened in this area of immunology, beginning with Jacques Miller's report from 1961-62. He had the very first reports out on neonatally thymectomized mice. And if you recall, when you did that, and you took the thymus out right after birth, and then let the mice just run around in the cages and so forth, they looked fine for about a month. But in the second month, they started becoming runted. They stopped gaining weight. They did the same thing that these IL-2 knockout mice did. And then the next thing that happened in the business was the IL-2 knockout mouse 30 years later, from 1961 to 1991. And then the next important thing that happened was in 1982, sort of in between in these 30 years, Shimon Sakaguchi from Kyoto University in Japan had reported in 1982, he'd sort of followed up 20 years after Jacques Miller, and he studied neonatally thymectomized mice. Jacques Miller was interested in the first month of those mice, and they were clearly immunodeficient when they didn't have any thymus, and that's what he wanted to establish. The second month, he reported it, but he didn't follow that up. And Sakaguchi did in 1982. He started studying nude mice and also neonatally thymectomized mice. And he reported a similar phenomenon when he tried to replenish the T cells, the peripheral T cells in these mice. And then 13 years later, in 1995, he honed down on a subset of T cells that were IL-2 receptor positive, alpha chain positive, CD4s, as well as CD8s. That was the beginning of the regulatory T cell. and then we get into the whole business, and that's what this chapter gets into, of the co-stimulatory molecules, the co-inhibitory molecules, CTLA4, PD1, and so forth, and the mechanism of how regulatory T cells regulate, how they suppress immunity. And this is part of the negative, this is the major part of the negative feedback loop in the IL-2 story, because CTLA4 is an IL-2 induced gene, as is PD1, and so forth. So you might check out this chat, buy the book, supplement my retirement income, and check it out. So that's the end of this story. And thanks for watching. And don't forget to like the video, click on the bell, and check out the social media sites. Check out the papers on my website. Thanks.